Yeah, good afternoon, everybody. Thank you for, first of all, inviting me to give this talk. And for those of you that are joining online now, and for those of you that may be watching this recording later. So I'm a lecturer at the University of Leicester. I've been here now almost 3 years. And I'll be telling you today a bit about some of the research that my group is doing, a little bit about each of the different research areas that we're looking into. So yeah, if anybody has any interests or any questions, please do get in touch afterwards, or obviously ask questions at the end of this talk. So my group largely uses cryo-EM and structural biology to look at DNA repair mechanisms and other biological processes. So just to give you an outline of what my talk will consist of today, I'll first of all give you a bit of background to the particular DNA repair mechanism that my group look at, and a bit of basics to cryo-EM for those of you that maybe don't know so much about cryo-electron microscopy. I'm then going to talk about my main research area, which is DNA repair in humans. And then my second research area is DNA repair in bacteria, specifically Mycobacterium tuberculosis, and tell you a bit about how cryo-EM has really helped us to understand these mechanisms further. And then the third research area is not DNA repair. It's more just looking at general drug activation and resistance in MTB. And with each of these three research areas, I'm just going to talk about the application and current and future therapeutics for each of these. So as I said, my research area 1, which is my main research area, is DNA repair in humans. So as I'm sure many of you know already, DNA repair is essential for survival of all kingdoms of life. So the problems come when you get mutations or unrepaired DNA, and this can be a really big problem because it can then lead to cancer, diseases, and aging. So your DNA can be broken in many different ways, and you can have lots of different types of DNA damage. You can get single-strand breaks, you can get mismatches, but perhaps the most dangerous and critical type of damage you can get to your DNA is when you get a break across both strands of the DNA. And these are called DNA double-strand breaks. And there's two main mechanisms that repair these types of breaks, but the main mechanism, and the one which my group works on, is called non-homologous end joining, or NHEJ. So over the past probably 20 to 30 decades or so, or 20 to 30 years or so, should I say, people have thought that the traditional mechanism of NHEJ is very simple, that it occurs in 3 main linear steps. And I'm now just going to show you just the cartoon depiction of how this mechanism occurs. And just to say, throughout the rest of my talk, there'll probably be some images of these cartoon depictions at the top right-hand corner of the presentation. So if you get confused which proteins and complexes I'm talking about, hopefully it'll be there to remind you. So it's thought that these double-strand breaks can occur through ionizing radiation, reactive oxygen species, chemotherapies, chemotherapeutics, that causes the double-strand break. The first step in repairing this is called synapsis, where a protein called Ku, shown here in kind of the orange and green, recognizes the double-strand break and binds onto it. It then recruits this larger kinase called DNA-PKcs, shown there in gray with DNA. Okay. The DNA ends sometimes need to be processed by nucleases or polymerases before finally a ligase, shown here in red with these scaffolding proteins XLF, XRCC4, then ligate the DNA together again. So these are the core or canonical proteins involved in NHEJ. But of course, over the years, people have identified many accessory and regulatory proteins which are also involved in this mechanism. So my group mostly uses structural biology to understand the molecular details of this particular mechanism. And we want to be able to visualize these proteins and understand exactly how they're kind of coming together and repairing DNA breaks. So how can we visualize the structure of these proteins? So I thought I'd just put this kind of fairly basic slide in here just to kind of put it into a bit of context. So if we think about what we can actually see with our eyes, we should be able to see each other, objects around us. But to look at greater detail at cells and organelles, you might use light microscopy. But to get the detail that we're interested in, so how these enzymes are working, their residues, amino acids, DNA, we need to use electron microscopy. And so that's where electron microscopy comes from. And it's because the electron wavelengths are higher energy than visible light, so it's able to get this more detail, this better detail of these proteins and complexes. The cryo element of the cryo-EM is because of it's being cold, it protects your samples. And the resolution revolution in cryo-EM in around 2014 was due to better detectors, better software, and so we're able to process our data and be able to attain higher resolution images and be able to see what our proteins are looking like. So just for those of you that don't know too much about cryo-EM, what we do is we first of all get our protein or complex. We make our samples in E. coli or insect cells, and sometimes we actually endogenously purify our proteins. You then place it onto a cryo-EM grid, which is then frozen into liquid ethane, which creates a vitreous ice. And then you put it into one of these cryo-electron microscopes. So this is one kind of similar to the one we have here in Leicester. They look just kind of like a big black box, really. But this one is actually one that's in Cambridge. And just to show you a video, really just showing you how complicated it is inside. And this is somebody just loading some cryo-EM grids into the microscope. And it's quite nice. This is the glacios they have there. You can actually see kind of the microscope and how it's all working inside. So if we then zoom in, if we then look at our grid under the microscope, we can see lots and lots of squares. These are the squares all over the microscope. If we zoom in on one of those squares, we should be able to see lots and lots of holes. And if we zoom in on one of those holes, we then hope to see our protein particles that are frozen. So how do we then reconstruct a 3D protein image from these many, many different images, or we call micrographs, showing our protein particles? So we use particular software that first of all picks these protein particles and complexes and sorts them into 2D classes. So these are 2D projections around your particular protein or complex. And you want lots of different projections of it to build up a good 3D reconstruction. So you then combine these 2D classes that builds up the 3D reconstruction. And from that, if your data is good enough, you should be able to get a cryo-EM map. And as I said, if you get good enough resolution, you should be able to build into this cryo-EM map then your particular amino acids and your DNA sequence. And then you should be able to get a lot of information about how these enzymes and proteins are functioning. So going back to my particular biological question, which is the NHEJ mechanism, we first started by looking at this step of the reaction, which is called synapsis. And this contains Ku, DNA-PKcs, and DNA, which together form a holoenzyme complex called DNA-PK. So I'm now just going to show you lots of cryo-EM structures that we've managed to obtain over the last 6 years or so. We do a lot of biochemistry, biophysical techniques as well, but just to kind of show you the power of cryo-EM and some of the information that we've managed to obtain from it so far. So the first structure we obtained was of Ku80. This is the first protein to recognize the double-strand break. So this is what Ku80 looks like. Ku70 here is shown in the blue, and Ku80 in the green. It's a heterodimer, and it kind of has this central hole, which is where the protein can bind DNA with a very, very high affinity. So you kind of get DNA binding through the central part there. And we now have the structure also with DNA bound. It's interesting to say, actually, that this structure is remarkably similar to the X-ray structure solved back in 2001. So we didn't see anything too different. What was interesting with all the structures we've solved with Ku70 is that we don't generally see the C-terminal region of either Ku70 or Ku80, which I'll tell you more about in a moment. We then went on and we improved the resolution of DNA-PKcs. This is this very large kinase. It's one of the largest single polypeptide chain proteins that there is, of about 4,100 amino acids. It's broken up into these 4 main domains: the N-terminal arm in purple, the circular cradle in this kind of reddy brown, and the kinase up here in yellow, which is important in phosphorylating itself and downstream proteins, and that's how the kind of mechanism works. And the improved resolution was really important because you can kind of visually see how many alpha helices are kind of making up this protein. It was really difficult to kind of register the particular amino acids in the entire sequence. And this protein is actually really important in terms of drug binding, inhibitors, and I'll go on to say a bit more about that in a moment. So the understanding and knowing how the residues were actually arranged was really important. And what's interesting actually is a lot of people always think kind of with structures, it's very static. But just to say that even in our structure of just DNA-PKcs on its own, we start to see some flexibility. So we can see the N-terminal arm moving up towards this head region, even within just kind of this data set. So we then went on and we wanted to get a structure of the entire holoenzyme complex. So this is now DNA-PKcs, Ku, and DNA. And after this kind of cut a long story short, a lot of optimization, we managed to get the structure of DNA-PK. So this is the structure. So DNA-PKcs is actually shown in gray here, with the N-terminal arm highlighted in purple and the kinase domain in the blue. And Ku80 now in the orange and green with DNA is kind of docked onto the side of it. And so if you compare the structure of DNA-PKcs on its own to when Ku80 is bound, we see this really big lifting up of the N-terminal arm. And just to show you kind of a video of this, this is a transition between DNA-PK as the holoenzyme complex and DNA-PKcs. And you can see kind of how the structure's breathing and it's allowing for Ku and DNA to dock onto the side of it. So these structures were kind of what we had already expected. It had kind of already been solved through crystallography or cryo-EM previously, and we just kind of improved the resolution. What we didn't expect to see in our data sets was that we started to see the vague image of a dimer. As you can see here, this is one PK, this is another. And through various rounds of processing and refinement, we were then very surprised and amazingly excited to see that there was in fact a dimer of DNA-PK. So this had kind of been hypothesized because you can understand that if the DNA is broken, that you don't want those DNA ends to kind of float away and maybe be joined in some random manner, and that could be, you know, really dangerous for the cell. But people have never actually seen that DNA-PK can in fact hold these DNA ends together in such a way. So this is our dimer of DNA-PK shown in gray compared to the monomer in yellow. And our resolution was just about good enough that we could actually dock in two DNA-PKs. So here is one PK with the other rotated by about 90 degrees in relation to the other one. And what was surprising really was that we started to see density that was there in the dimer that was not there in the monomer. And so we wondered what this extra density was. And what it actually turned out to be was the C-terminal region of Ku80 that we never usually see on Ku80 on its own. And so we could actually see density for this linker region leading up to this globular part of Ku80 and then a linker leading to the C-terminal alpha helix. And what was even more interesting was this C-terminal alpha helix. What it's actually doing is going from one DNA-PK proteomer over to the other one in a domain swap-like mechanism. And this C-terminal alpha helix has been shown in vivo to be really important for DNA repair. And so we were seeing then for our structures why this was the case. So this kind of opened our mind really to then thinking, you know, perhaps this dimer is acting as a stage. You know, maybe then all these other proteins involved in the repair mechanism can actually dock onto this and are then involved in actually joining the DNA ends together. So we then went to later steps of the reaction or mechanism, and we started looking at ligation. So ligation includes these proteins, the ligase, shown here in red, which is important in actually ligating the DNA ends together. It's made up of these BRCT tandem repeats, and they're flexibly tethered to the catalytic domain of ligase. And they have a tight interaction with this protein called XRCC4, shown in this kind of bluey color, which has this long stalk and head domain. And in the same family as XRCC4 is a protein called XLF, which is shown here in pink. You can see it's kind of in the same family because it has this stalk region and this head domain. And so what we did with our optimized cryo methodology was that we then added in these proteins. And with a lot of data processing and a lot of kind of optimization, we managed to reveal that there was in fact another type of DNA-PK dimer. So this dimer is actually different from the Ku80 mediated dimer. We call this XLF mediated dimer because it's this pink protein in the middle here that's actually kind of at the interface of the dimer. The DNA-PKcs interface is different from what I showed you a moment ago. And this is a large supercomplex, you know, it's 1.6, 1.7 megadaltons now. And what's interesting is that XLF is then interacting through its head domain with XRCC4. And then this is interacting with the BRCT tandem repeats of ligase, which interact with Ku80. And as I say, it's mediated by XLF, so we call it the XLF dimer. But in this structure, although we added in full-length ligase, we do not see the catalytic domain of ligase. And that is maybe, you know, what we would expect, because you can see the DNA ends. There's one DNA here and one DNA over here. They're not at the point of when they can be ligated together. So the ligase, there is no role for it in this particular complex. So we're looking at kind of pre-ligated or pre-ligation steps of the particular mechanism. So I've so far showed you two different types of dimers, one mediated by Ku80, the green protein, and one mediated by XLF, the pink protein. And structurally, these two dimeric assemblies are actually really quite different. But what was amazing to us was when we actually looked at the distance between the DNA ends, they were almost identical in both these structural assemblies. And through some really nice kind of single molecule experiments by other groups, they showed that during the DNA repair mechanism, it goes via a long-range synaptic complex before a short-range complex where the DNA ends can be ligated. So we then have called these two long-range synaptic complexes because they're holding the DNA ends before they can actually then be joined together. And so with a collaborator, Professor Kathy Meek at Michigan State University, we tried to, of course, disrupt these dimers to see if there was any effect in recombination or in repair. And you can just see here, the wild type is in the white and all the colors are different mutants of these particular interfaces. And you can see there is a dramatic effect on efficient DNA repair. So these dimers are, in fact, actually important. So I've now shown you two different types of dimers, and there's lots of accessory and interacting proteins during this particular mechanism. So what we then wanted to figure out is, how do some of these other accessory proteins actually interact with these dimers? Do they favor one dimer over the other, or do they have different types of DNA end configuration which affects dimeric assembly? And just to tell you a little bit of recent work that we've been doing on one of the accessory proteins or regulatory proteins, and that's PAXX. So just to tell you a little bit of about PAXX, this is shown here in kind of the blue cyan color. And again, it's in the same family as XLF and XRCC4. You can see its stalk region is a lot shorter, but it's got this head domain. And interestingly, XLF and PAXX have functional redundancy. If you delete both of these, then it's synthetically lethal in mice. And so kind of the interplay between XLF and PAXX was, not completely understood. And so what we did is we first of all took Ku80 on its own and added in PAXX, or a particular peptide of PAXX. And what we identified, this is a Ku binding motif of PAXX, was we got two structures, one of Ku80, where it looks like this, and this particular domain of Ku70 called the VWA domain is actually in its closed state. And then we got a structure where we can actually see density for the peptide of PAXX bound. And now what's happened is this VWA domain has really opened up to allow for PAXX binding. And if we just zoom in here, we can kind of then nicely register this particular peptide and look at interaction interfaces. And in fact, we did some really nice mutational studies of this, showing the specific residues which are actually interacting with Ku80 from PAXX. And so, yeah, we can see this really nice opening, and this video just kind of shows it to you. This is with and without PAXX binding, how the protein is kind of really opening up to accommodate for PAXX. And we did this in collaboration with Professor Charbonnier in France, and he did some X-ray crystallography experiments as well, showing the exact same thing of PAXX binding. And AlphaFold actually also confirmed it once we had told it the particular region that we were looking at. So we then went beyond this. We wanted to see how PAXX actually interacts with the long-range complexes that we had established. So we took DNA-PK and ligase, XRCC4 and PAXX, and this is what the data was that we were able to generate. So we still saw the Ku80 mediated dimer, but we could now see density in the middle here. So we assumed this must only be PAXX because this was the only extra thing that we'd added in. And to confirm this, we looked in the VWA pocket of Ku70, and indeed we could see density for the peptide of PAXX bound. So although the density in the middle was always kind of very poor resolution, that the peptide was actually very, very clear. And what this is showing is that PAXX can actually stabilize the Ku80 mediated dimer. Okay, and it's in the center, which we never saw with XLF. So we then decided to add in XLF. can both of these proteins bind at the same time? You know, they kind of have this functional redundancy, but they're both synthetically lethal. What does it look like? And so we got these structures. We got the Ku80 mediated dimer, where we can see central density for PAXX. We can also see XLF bounds. However, it's not in the center this time like it is in the XLF dimer. It's kind of more towards the side. And we could also see extra density in the XLF mediated dimer, showing that PAXX can also bind to this. And to prove that they could both bind at the same time, again, we looked in the pockets of the VWA domains, and we could actually see density for XLF and PAXX in both of these, showing that they can both bind. So we kind of concluded, and there was lots of other work in this kind of publication that we published just last year, was that PAXX and XLF can both contribute to long-range synaptic complexes. We think that the structural redundancy maintains formation of at least one of these dimers if you remove PAXX or XLF, because we can see XLF is important for this dimer and PAXX is very important for this dimer. So we could kind of start to understand what the interplay between those two proteins is and why they're both really important for repair. So we've got this kind of working cryo-EM mechanism now where we know the double strand breaks. We know Ku80 is the first protein to recognize a double strand break. And we know we have these two dimeric long-range synaptic complexes that we're still trying to figure out, you know, which one forms when. Is it kind of DNA dependent? Is it protein dependent? We know that proteins like PAXX can kind of stabilize some of them over the other ones. And then what is known is that DNA-PKcs is phosphorylated. It's removed from the complex. And then what you get is a short-range complex. And in this short-range complex, the catalytic domain of ligase is in fact now present and can then ligate the DNA ends together. But these kind of intermediate steps in between are still kind of fairly unknown. So just to show you some of the other work that we've been doing, we've been looking at kind of IP6 binding, which is a small molecule that binds to Ku80 and increases the binding affinity for DNA. We've looked at drugs binding to DNA-PKcs, which I'll tell you a bit more about in a moment, as well as I just showed you the work on PAXX, and we've been looking at some of the polymerases and nucleases. And just to say that we also think not only DNA-PK dimers exist, but higher order assemblies, and we've started to identify that perhaps trimers, tetramers, and I'll show you in a moment a bit about filaments and how this can be in response to multiple double-strand breaks. So I started my talk by showing you the traditional NHEJ mechanism, which has been thought to occur over the past, quite a few years or so. And from our cryo-EM data, we now believe it looks a little bit more like this. It's quite complicated. It's opened up a load more questions, and some of them, we hope to answer. So, what are the intermediate steps? How are these proteins kind of transitioning from one to the other? What are the end processing enzymes doing? And is that kind of DNA end configuration dependent? Is there kind of a branched pathway depending on the type of damage, depending on the type of DNA end configuration? How do all these proteins access the double strand break when it's packaged in nucleosomes in chromatin? And, do these filaments exist of DNA-PK? And what's the time course of this? How long does this process take to happen? And importantly, we're kind of moving even beyond using single particle cryo-EM, and we want to start to look at cryo-electron tomography, which is looking actually inside the cell, so in situ, how these proteins are actually repairing it by taking a cell and kind of taking slices of that. So just to show you quite briefly, really, some of the ways in which we're hoping to do that. So the first thing is we want to kind of map every single structural step of this. And to do that, we have to kind of trap structural intermediates. And so we have different ways in which we can do it. And just to show you some of them here. So as I just showed you, we know that we can add in accessory proteins such as PAXX. We can drive different branched pathways of the mechanism. We can activate the kinase DNA-PKcs with ATP. And we can also trap structural intermediates using specific DNA. We can also inhibit the protein with drugs and non-hydrolyzable ATP. And very recently, we've shown that actually the DNA-PK dimers do favor certain DNA end configurations. And we also have mutants of some of the enzymes involved in the mechanism, and we do kind of complementary biochemistry repair assays along with this. And very recently, we've started a collaboration at the University of Leeds, and what they have there is a time-resolved setup where they can actually take, for example, DNA-PK, add in ATP, freeze grids within the millisecond timescale, and actually see what's going on with that particular complex. So that would allow us to kind of figure out how long this is actually all happening and taking. The second thing is to understand on a biological context how these are accessing double-strand breaks. So we see in our cryo-EM data these filaments of DNA-PK, but do they actually exist inside the cell? And we've started to see that certain proteins can actually stabilize this filament. So is it important for multiple double-strand breaks? We also, with collaborators, do functional in vivo experiments. So we look at our structures and we, kind of disrupt these interfaces and have a look at how that affects, recruitment to DNA damage sites, DNA repair, and those kind of experiments. And we also want to look at how the proteins access the double-strand breaks when they're packaged within nucleosomes and chromatin. And a PhD student of mine is just working on looking at these NHEJ proteins binding to nucleosomes. And we have some recent data showing that Ku can actually interact with the nucleosomes. So how does it access the double-strand break? And as I said, we're really looking kind of forward in kind of structural biology and trying to think about how we can use cryo-electron tomography to look actually at how this is happening inside the cell. So there's been recent papers, and this is another group, not our group, that have looked at nucleosomes and chromatin packaging using cryo-electron tomography. And so DNA-PK is really abundant actually in the nucleus. So we're really hopeful that we can kind of cause DNA damage to cells and then visualize it and process the data and see what's actually happening. And so the application of this particular research area is that it's, chemoradiotherapy damages DNA in cells. So NHEJ, because it's repairing DNA, is actually quite detrimental to cancer treatments. So there's been a lot of work on trying to inhibit NHEJ to enable and to help with these particular treatments. And so there's several drugs already which target DNA-PKcs in the ATP binding site, and we've done a little bit of cryo-EM to show how some of these molecules actually bind. And we're kind of looking at new ones all the time to see if we can kind of optimize them, make them more soluble and more specific. And there has been some work on targeting some of the other NHEJ proteins as well as the other protein-protein interfaces as well. And that's still kind of ongoing. And just to say, we're collaborating with a company in France called Valeria Therapeutics. And what they have is they have DNA mimics. So these are DNA molecules that actually allow for the NHEJ machinery to bind to the mimic rather than the cellular DNA. And therefore, that then leads to cellular death. And so we've done a little bit of structural work, but this is still kind of ongoing, trying to look at how these DNA mimics actually bind to DNA-PK. And we've seen with one of them how it can actually activate the enzyme. And the idea is that if we can kind of trap all these structural intermediates and map this entire mechanism, then hopefully that will open up, the avenue for development of more specific therapeutics and targeting. So that was the first research area. And just to say, the other two are not quite as involved and in depth. But I just wanted to mention them just to kind of let you know what, the group is doing and to give you a bit of an idea about what research is going on. So the second research area is still DNA repair, but it's now in bacteria. And specifically, we've been looking at Mycobacterium tuberculosis. So NHEJ in bacteria is simpler than in humans, as you could imagine. So you still get the DNA double-strand break. Ku recognizes a double-strand break, but in bacteria, it's a homodimer. So it's simpler, it's smaller. Then what happens is it recruits Ligd, and Ligd is a multifunctional enzyme, which I'll show you in a moment. And then it then repairs the DNA. So NHEJ is simpler. The ligase, so this is Ku here, and this is Ligd. Ligd is actually made up of a ligase domain, a nuclease domain, and a polymerase domain. So it's able to do, a whole host of different mechanisms and functions that actually in humans multiple proteins do. And Ku is a homodimer in bacteria and tuberculosis, and you can see here in pink it's much smaller than in humans. So humans have got these VWA domains that we just looked at, whereas in MTB it doesn't have them anymore. And maybe that's because it doesn't need to bind these other accessory proteins like it does in humans. And looking at tuberculosis, is hugely important, and I'll tell you a bit more about that in a moment. But it affects 1/4 of the world population still. It's there's a lot of drug resistance being prevalent. So understanding kind of these fundamental mechanisms and kind of the differences with humans as well is really important. So what we've started doing so far with this is we've taken Ku from MTB, and we've put them onto grids with particular DNA. And just to show you very briefly, we have amazingly actually revealed that Ku from MTB actually forms a filament. So this is not what we expected. We assumed that we would have a structure of Ku bound to DNA, but in fact, we get kind of these multiple homodimers of Kus. So one in purple, one in green, and one in blue. And you can see a bit of the DNA there, and there is a bit up here as well, which I don't think we've quite modeled in yet. So this doesn't happen whenever we've used the human Ku7080. So we're trying to understand why this happens. Is this important for DNA repair in bacteria? And, is the interface between these Kus actually maybe, some kind of target that is unique to bacteria that isn't for humans? And so the application therapeutic for this is that although NHEJ isn't important, it isn't essential for the bacteria, it is important during latency. So could this be some kind of antimicrobial target? And we're kind of trying to identify key differences and understand this filament further, add in different types of DNA, and starting now to add in Lig-D. How does the Lig-D enzyme actually perform multiple functions that in humans is done by many different proteins? This is just the alpha fold structure of Ku-MTB with Lig-D. Ku-MTB is shown there in the purpley pink and Lig-D in the blue. And there is supposed to be a specific interaction between the C-terminal part of Ku with Lig-D. But yeah, this still needs more data and more information to help us understand more about what's actually happening in DNA repair in MTB. And the final research area that I just wanted to speak to you today about is moving away from DNA repair. So we'll forget that DNA repair is a thing at the moment. And this is actually just thinking about drug activation and resistance in MTB generally. Okay, so just to again, reiterate the problem with tuberculosis, that annually 1.3 million people die, and that's from the WHO 2022 data, and a lot of this is due to drug resistance. So we've been looking at a couple of enzymes which activate particular prodrugs. The prodrugs are isoniazide and ethanamide, which isoniazide is a first-line prodrug and ethanamide is used for multi-drug resistance. And it's known that mutations in these particular enzymes then leads to drug resistance. And even though, isoniazide particularly has been around for a long, long time, the location of drug binding is actually still not known. So CatG is a catalase peroxidase. It contains a heme cofactor, and this is what activates isoniazide. So this is the protein of CatG here. And this is actually one of our experimentally determined structures, should I say, that we solved a few years ago. And ethanamide, this is the alpha fold structure, because until very recently there was no experimental structure. This enzyme is supposed to activate ethanamide. So what we've been doing so far is we first wanted to try to identify where isoniazide binds on CatG. There's been some kind of crystal structures on this, and it's all been kind of a little bit unknown where it binds. And so we did some experiments and we tried to identify it. I have to say it was very difficult. It's a very small molecule and there was a lot of dynamic kind of binding, but perhaps the most obvious binding site was at site 1, which is where there's a particular resistance mutation, S315, which we think perhaps this is where isoniazide is most likely to bind. We have then done some resistance mutation experiments with CatG. So there's particular clinical resistance mutations. We looked at W107R and T275P. They're kind of fairly near to the heme binding site. And what we actually found is that these particular resistance mutations, what they do is they actually then affect heme binding. So they prevent heme binding to the protein and retaining the heme. And therefore, if it doesn't have this heme cofactor, it can't actually activate the prodrug. It kind of can't turn it over. So we did a kind of a small study on this, just trying to look at how these particular mutants affected heme binding. And even when you kind of loaded the protein with this particular heme cofactor, it was never retained. And so we're still now trying to look at other resistance mutations and determine whether this is indeed, how this is actually functioning. And just to kind of very lastly show you actually, we have some very recent data on ETHA. This is the enzyme which actually activates ethanamide. And we have showed that, again, it forms higher order assemblies. We have noted that ETHA can actually form two different types of hexamers. One that we say looks like a clover, one that looks like a donut, and it can actually form a filament. Now, we don't know why it's forming these two different types of hexamers and whether one is kind of an inactive or an active state. I should say that at these hexamer interfaces, we see some areas of extra density that we don't know what these areas of extra density are. So if anybody would ever have any, ideas about what this could be, I should have maybe shown an image of what this area of extra density is. It's kind of at the interface of the trimer of this hexamer, and there's lots of lysines, arginines, which are interacting with this area of extra density. So we think it's some kind of maybe phosphate, some kind of lipid. We're a little bit unsure. So we're thinking about trying to do some mass spec to try to identify it. And we're now collecting data with looking at drug binding, and NADPH as well, because this protein has FAD cofactor and NADPH as well. And we want to know, is this filament biologically relevant, which is kind of what we always want to know. And yeah, just to summarize really that, the aim of my research is to understand the fundamental mechanism of NHEJ in humans and bacteria, and understand drug resistance in MTB in order to generate new ways of developing drugs in the future. And just to thank many people. The NHEJ project started in the University of Cambridge in Tom Blundell with the cryo-EM facility and people there. And now this is my group at the University of Leicester, who are fantastic, and everybody there has been great since I started, a few years ago now, and the cryo-EM facility there. And these are some of the collaborators that have been involved with the work that I just produced and just presented to you. So thank you very much. Hopefully I didn't whiz through that too quickly. And if you have any questions, please ask. If not, please get in touch and drop me an email. Thank you very much.